thank you very much for the invitation, first of all. And after this exciting start, I'm going to take you to an even more exciting journey. And we're going to eavesdrop on the metabolic crosstalk that takes place in algal blooms in the ocean. As you can guess by the title, it's a very different environment than we had in the last days. And I also spent the last days to understand what might be the analogies between the research that I'm doing and the research that you are doing. For the beginning, where I'm situated, I'm from the lab of Asaf Vardi at the Weizmann Institute of Science. This is our department, and this is Asaf. And as you can, we are studying marine phytoplankton, the microbial interactions in marine phytoplankton chemical signaling. And as you can guess by the images, a lot of our work is taking place in the marine environment. Still, model systems are definitely on demand in our microbial world in the ocean. There are only a few that are genetically modifiable. The ocean is our largest habitat. 71% of the Earth are covered by the ocean. And it's not only the origin of life, but it's also an important climate regulator. And it's responsible for 50% of the oxygen that we breathe. So 50% of the photosynthesis that takes place in the ocean is coming, I'm distracted a little bit. 50% of the photosynthesis takes place in the ocean. So every second breath that you take is originating from small little microalgae in the ocean that are usually not visible without a microscope, but they can form these large blooms that are even visible from space by satellite images. The life in the ocean in general is dominated by microbes. 2/3 of the biomass in the ocean are microbial cells, like protists and bacteria, archaea. And the most abundant entity are actually viruses. This is a DNA-stained drop of seawater, and it has been estimated that there are 100 million times more viruses in the ocean than there are stars in the universe. In focus of my research are these algal blooms that I already mentioned. These are essentially events of high proliferation and concentration of phytoplankton cells in the ocean. They can come in different flavors, like coccolithophore blooms, cyanobacterial blooms, dinoflagellate blooms, and diatom blooms, usually along the coast, and therefore also affect very often our human life. So it's a combination of single cells that are forming a multicellular entity, as you can see it from your point of view. And we are interested in the development of these blooms. So that's the developmental point or aspect of our study. And what you can see here is a model from the oceans and the occurrence of 4 of the major algal groups that we have: diatoms, flagellates, cyanobacteria, 2 different types of cyanobacteria across the ocean. And as you can see, there is it's highly dynamic in their occurrence. They are mainly structured by the seasons, of course, in their occurrence, but also by other environmental parameters like ocean currents, nutrient availability. And this development is reoccurring every year, and we are studying what's helping in the bloom development, bloom demise, and we're going to look in this a bit more in detail later. There's also a very strong flow of energy and matter in algal blooms, to take the second analogy to the workshop. Algal cells are the ones that are taking carbon dioxide, are fixing using sunlight this carbon into a plethora of different organic molecules. As I said earlier, 50% of the global primary productivity is coming from these algal blooms. However, what's different to the land is that they kind of represent only 1% of the biomass of plants on land, which means that the standing stock is very low. So there's a lot of production where there's no biomass kept essentially in the oceans. And this means there's a strong flux of carbon going through these blooms. Where does it go? On the one side, we have a lot of grazing taking place. The whole marine food web is based on algal photosynthesis. A second major zinc is the release of dissolved metabolites, and this is a bit more important for my work, of algal cells. So the active or passive release of metabolites from cells into the dissolved organic matter pool in the ocean. And that's an important one, as it's the food source essentially for all the microbial heterotrophic world, like bacteria, that are respiring this covering. And another entity that I already mentioned that's important in my work is viruses. They are diverting, or interesting, as they are diverting essentially carbon from this particulate phase into the dissolved phase and fuel the marine microbiome. So there is a lot of energy and carbon circulating through the ocean essentially, and I want to visualize this with one more example that I want to kind of connect to a presentation from yesterday, how this can happen in practice. We yesterday heard about on-carbon metabolism, and algal cells are very strong producers of methylated small metabolites. On the left, you can see DMSP. It's one of the most abundant metabolites that algae produce. And then two kind of degradation products of it. And it has recently been shown that these methylated compounds can lead to a lag phase shortening in the bacteria that are associated to algae as a kind of host microbiome. So the more methyl groups algae provide, the shorter is the lag phase of the bacteria to kind of get out of stationary phase. And that's only one compound that we kind of was here looked at, and different methylation patterns, but there are actually a huge diversity of these compounds present in algal cells: choline, carnitine, betaine, homarine, trigonelline. Like they come in all different kinds of flavors, and that's one way of how it looks like algae can share their energy and the carbon that they preserve with the algal microbiome that they attract or recruit. In the same study, it has also been shown that the methyl groups in here are not only a carbon and the energy source, but that they are also signaling in this context. So these metabolites can also lead to a flow of information in these microbial communities. And in the last years, we have discovered and identified multiple of these infochemicals, as we call them, and they can mediate different types of interactions between algae and parasites, between algae and grazers, between bacteria and algae, as we just looked at, but also to structure algal communities themselves. I want to give 3 examples a bit more detail that might be more connected to developmental aspects, and you find analogies in. So on the left, we see a macroalgae actually. So let's start not from a single cell example, but from a macroalgae that is common in coastal areas, Ulva mutabilis. And on top, we see a metabolite called tallosine. This is not produced by Ulva, but it's produced by a bacterium called Maribacter, which lives in close association to this algae. And it is important for the cell differentiation. So in the absence of this bacterium in the coast, this algae would not be able to form this morphology. Another example is shown in the middle, where, but a different type of interaction. Huxley's form is a single-celled organism. It forms large blooms, and this molecule on top, HHQ, is a form of sensing signal produced by bacteria that are associated to the microbiome of these blooms. So bacteria use it mainly to help their own proliferation or community population size, but it also leads to the cell arrest in the associated host algae. And a third example how these microbial cells can interact and influence each other's growth would be the one on the right. This is a diatom chain. It's composed of single cells that are elongating in this chain line pattern. And on the metabolite here shown on top is called copperpotamide. It's produced and released from the grazer of this diatom. And when the algae experience it, they will shorten their cell length in order to get too small to be eaten. So these are just 3 maybe examples how these microbes are actually quite heavily reacting to each other. If there would be something like a dictionary called Chemical Vocabulary of Marine Algae, which is not existing yet, it would be still quite empty. So there are only a few of these cases known, and where we also were able to identify the metabolites that are involved. We have way more phenotypes described, where still the metabolite or the chemical cue has not been identified yet. So that's this ongoing work, and that's also essentially the main research interest of me, to expand this vocabulary of the chemical language that mediates microbial interactions and algal blooms and supports the algal bloom development and bloom demise. One of the main challenges in our work is that microbial interactions and the language that mediates this interaction is context dependent. So if you are starting to assess these things that are happen in the environment repeatedly, you try to bring them in the lab. Very often you lose most of the phenotypes because of the reduced complexity. And the way that we are trying to work with it is that we are doing a lot of environmental work. So I'm applying environmental metabolomics to study these interactions in the natural environment, in algal blooms, for example, directly in the open ocean. But we also have ways to reduce a bit the complexity using mesocosm experiments or synthetic communities. Just for the technical point of view, there's an additional challenge in the sense that most of the compounds we're looking at are highly diluted in the ocean. So when we want to look at metabolites in the sea, we usually take samples, we take out the particulate phase, we use the filtrates, which are comprising all these metabolites, and extracting them to increase the concentration and be able to measure the metabolites of interest with a different range of polarities. How does this look like? This is probably the smallest way of how I had to work so far, which is on the schooner Tara, where I got essentially the space of an A4 page to set up my experimental technology. And what in the back essentially are two samples that we took from one particular station in the coast of Patagonia. And then the water is, after filtration, led over the solid phase extractions. It's speed dependent, and on the sorbent phase, the metabolites are captured. And then like this, we can later analyze them in the lab. In some cases, it looks more like that. When you have a bit more space and we can take more replicates. This was in a field station. But again, as essentially most of the work we're doing is not standardized, we are having to develop a lot of the methodology still in this field to improve measurement of metabolites. I want to give you now 4 examples how the metabolism of an algal cell can affect interactions with its microbial community. In the first case, we will look at algae called Phaeocystis. It's a single-celled algae that can be found both in symbiosis, as shown on this image. It can form colonies that are held together by repeated cell division and a polysaccharide matrix. And if these blooms, this is the form in which it mostly blooms, is demise and it reaches the shore, it can foam up essentially, and this is how we usually experience it as humans. So it has interesting life stages, and the question during my PhD was if the cell metabolism of this algae is kind of constant. So we looked on metabolic plasticity both in the lab and in the field. We did lab experiments to follow cultures through their growth. So they are having, in batch culture, exponential growth. They have a stationary phase, and the metabolism of the cell varies following essentially nutrient conditions and irradiance conditions. In the beginning, there's a lot of amino acids present in the cells, free amino acids. During the early stationary phase, there's an increase in sugar alcohols and saccharides. And in the late stationary phase, there's an increase in more fatty acids and sterols. And what was nice to see is that when we moved from lab conditions to natural conditions and sampled here a gradient between Norway and Svalbard, that's the natural habitat of these algae, and looked on different algal blooms in its natural context, we could find the same plasticity. That's essentially the environment that shapes algal cell composition. This can be reflected in, or it can affect the way that they are now interacting with their surrounding microbes. That's the next story I want to link it to. We are switching now the algae. Now we are looking on one that's called Emiliani Huxley. It also forms blooms. Here we see single cells. They have this beautiful little calcite skeleton around their cell, which also reflects the light strongly. And this is why we can easily trace it by satellite images. So this is a small little ship. This is a bloom of Emiliani Huxley. And it also can exist when this calcite is sediments. It also forms sediments essentially on the long term on geological time scales, as for example, the cliffs of Dover that are essentially composed of these algal blooms from the past. And this algae is surrounded by bacteria, different types of bacteria. One of them that has been isolated from its blooms is Sulfitobacter. Here you can see an algal cell and two of these bacteria attached to it after it lost the calcified skeleton. It's a bacterium that lives for a while in coexistence. When we co-culture essentially algae and bacteria, this bacterium, there is a period where there's no effect essentially on the growth of the algal cells. As soon as these algae are reaching stationary phase, the bacterium switches its behavior, it gets virulent, and this leads to the demise of the population. And this is mediated, as we could find out, by two metabolites. One of them is DMSP, which is shown here on the bottom. It's one of these one carbon metabolites that I mentioned already. In gray, you can see the concentration of DMSP in the cultural medium in the absence of the bacteria. So there is a certain amount of DMSP in low levels released into the water, but in the stationary phase, the cells get either way more leaky or produce more of it, so that the external concentrations of DMSP strongly increase. Meaning the bacterium gets exposed to this compound at the life stage of the algae, and that's the same time where they switch and respond to it by getting virulent and acting as a pathogenic bacterium. There's another compound that is released by the algae, which is called benzoate. Also, this one is released into the water. And interestingly, when we use both of them in combination, they can balance essentially the behavior of the bacterium towards the algae. We see here again a co-culture in gray with Emiliani Huxley and its bacterium in stationary phase. There is this switch that leads to the demise. If we are adding benzoate, there's no change in that. So it's not a compound that in that sense has either inhibiting or advancements for the algae itself or the bacterium. If we are adding DMSP, as I told you, this has acts as a signal for the bacterium to switch to virulence, meaning that the bloom demise happens earlier. As soon as we combine them, there's a balance. So benzoate can essentially prevent this pathogenic behavior of the bacterium for a little. So in this case, the metabolites that are produced, the metabolic state of the algal cell, represented and here narrowed down to just two different types of metabolites that are released by algal cells into the medium, affects the behavior of the algae that are occurring in its microbiome in nature. In the next part, I want to make it even more complex. So there is another entity that we know that occurs in the environment that I mentioned already, which is even more abundant than bacteria, which are viruses. And viruses have the potential to change the metabolism and lose the metabolites that are present around a cell by their own during infection. Algal viruses are giant viruses. That means that they are bringing hundreds of genes into the host cell that are transcribed during infection and change the host cell metabolism. We call this state virus cell. In the case of Emilia-Huxley, it's about 500 genes that are introduced, and different auxiliary metabolic genes are included, such as DNA polymerases, specific tRNases, fatty acid desaturases, and one that I want to highlight, which is a sphingolipid biosynthesis pathway, almost complete one. It's the only virus that we know today that encodes for sphingolipid metabolism. On the bottom, you see, by the way, a Emilia-Huxley cell and the viral progeny. Every black dot here is a viral particle. And what happens during infection is that the sphingolipid pathway of the algae itself, so also the algae produces these compounds. It has two specific ones that we usually use as biomarkers. One that is present in all the cells that you can find, and one that is having a different sugar head group, sialic acid, which makes it susceptible for viruses. Essentially, if this pathway is not present to produce SGSLs, the cells would not be infected by the virus. So that's essentially the one that's present in the host during infection. The virus replaces most of these genes by its own genes that are transcribed stronger, and this leads to a different glycosphingolipid in the end, shown here on the bottom. There's, for example, an additional hydroxyl group present in these glycosphingolipids, and it uses, instead of C16 during synthesis, C15. We use these different metabolites that are produced during infection as biomarkers that help us, because they are essentially only produced in this specific situation, that this algal cell and this specific virus are actually meeting and get infected. Otherwise, you would never find these compounds in the natural environment. So they're very, very specific biomarkers for an interaction, an active ongoing viral infection. They are also part of the viral membrane. That's the reason why they are produced. And interestingly, they also have a signal component, as they can trigger programmed cell death in neighboring cells. So viruses really have the potential to reshuffle metabolism of an algal cell. And the question in my postdoc was to look on the external impact of this. After the infection, usually this leads to cell lysis, to the release of virions, to the release of vesicles, but also to the release of this remodeled metabolites. For this, we went to work with natural blooms. In this case, a setup that's called a mesocosm. So we went in an area in Norway that is very often experiencing blooms. This is Bergen, and there's a marine biological station that belongs to the Bergen University. And here you can see a natural occurring bloom of Emilia-Huxley in 2020. And what we do in these setups is we went 1 year late, 2 years earlier, sorry, to this place, and we pumped the water from the fjord into 4 of these enclosures. So you make use of the natural complexity of the organisms that you expect to be present anyway already in the water. And then by adding nutrients, you can enhance and induce the bloom that would naturally happen anyway at a different time point maybe. So they are still having the whole complexity of the community. And here we sampled for 23 days with a bunch of people. And what's first of all interesting, what happens when you work with communities, there were 4 different enclosures. They don't behave the same. So here we can see the bloom of Emilia-Huxley in each of the bags. There's this increase in cell abundance that happens more or less at the same time in each of the bags. And then there's also a bloom demise that took place, strongest in the bag 4 in this case. And when we look on the presence of the virus in parallel, using the major capsid protein gene in this case, you can see that there's almost no detectable increase in this viral gene in the first case, where there's a strong increase of this gene marker in the last case. And as I told you, there is a lot of reshuffling happening in the metabolism. So if we are now switching on the metabolic level, we can see the same thing, that there is a strong production of this component, virus-induced glycosphingolipids. I will have to speed up a bit. So my question was, if we are not looking on the extracellular landscape, can we find metabolites that are related to this infection? It's very complex. There are tons of different metabolites and different patterns that are present. That's always a challenge we tackle. Most of them also are unidentified. I used this behavior to correlate now all the metabolites, and this led me to a subset of 20 metabolites that are all not known. They are all, when you compare them to libraries, not existing in libraries, unknown novel metabolites. But they follow the viral infection in the bags. And interestingly, the majority of those were halogenated. They contained dual halogenation of chlorine and iodine, which is very rare in nature. And if you think about bioactivity, most of these halogenated compounds are highly bioactive. As they are not present, and you can't, for example, buy them now in by Sigma, what we tried, I will skip on that. We are trying at the moment to kind of establish and reconstitute this in the lab. So we found after 2 years of Corona, a setup that allowed us to produce metabolites in the lab that also are having this dual halogenation upon viral infection of lab cultures. And what was interesting here to observe, and which also was a reason why it took us so long, usually you work with actinic cultures. In the absence of bacteria in the algal microbiome, which is the 2 bottom lines, there was no production of these chlorine iodine compounds. Only in the presence of the microbiome and in additional infection by the virus, you can have the same functionality under lab conditions. So the algal microbiome was necessary to reconstitute essentially the natural occurring chemical behavior. And here we are focusing right now on the biosynthesis of this structure, working on the structural elucidation and the ecological role. And I will finish with one thing. I will switch with another story, also my summary. But what I want to let you listen to in the end is that in the end, this maps that we are working on, metabolic maps that represent kind of all the complexity of metabolites that microbes are experiencing in nature, is a kind of fluctuating conversation that takes place that for us as humans is not audible because we don't talk biochemistry. I hooked up with a guy called Antoine Bertin during the Tara mission, and he used our metabolic map to make visible what we can't listen to, like the chemical communication that might take place during these algal blooms. So he used this data set essentially and made it audible. And that's part of these are the halogenated compounds that I showed you. Thank you for attention.